feel it. You are invested in these drug discovery projects, and you do get excited about them.
I'm happy to welcome Adam Field to the first episode of my podcast in 2021. Adam is a senior scientist at Sygnature Discovery, a UK-based contract research organization where he is responsible for many drug discovery projects, utilizing biophysical techniques for assay development. We first got acquainted as graduate students in San Diego, and on this episode we dive into his current role at Sygnature, his leap of faith to quit his PhD work, sustainability efforts from scientists, and some British sitcoms. Welcome to the podcast. Thank you very much for joining me today on what's been a way overdue of a podcast episode. The last one I have done was back in June of last year, and boy oh boy, a lot of things have changed since then. But not only things have changed since just 2020, it's been almost 3 years that we've had a chance to actually share a pint of Newcastle ale. Since our time in San Diego. Obviously, we both moved on from that city, and I think in the last 3 years, have your movie preferences or TV preferences evolved? And that's kind of the first icebreaker question I like to ask, and this is like a segment which I call Covideo Recommendation.
Oh, nice. Well, you'll be happy to hear that there's no shortage of TV I've been watching. It's it's one of the few activities that you can get away with during COVID times. So I've been catching up on quite a bit of film and TV. I would say my tastes have probably stayed similar. I'm not overly adventurous when it comes to film and TV, so sorry to disappoint.
I can relate. I mean, was one thing not adventurous, and the other thing is repeat viewings of the same thing, which is what I'm guilty of. Where do you fall in that category?
I very much do the repeat viewings. I catch up on old sitcoms and things like that. Keeps the mind happy and comfortable.
So old sitcom, is that a Friends or some other one, for example?
Oh, I'm talking more like British sitcoms, IT Crowd, The In-Betweeners, things like that. They I don't know, they have a nice nostalgic feeling, and those jokes don't get old, at least not for me.
So do you know what would be like the defining difference between a British sitcom versus American sitcom?
Oh, I'm not entirely sure. Other than the length of seasons.
Okay.
Which is, I think the British sitcoms, they tend to be you get 5 to 10 episodes that are just back to back, non-stop hilarity. Whereas the American sitcoms, they go on for a lot longer, so you get a bit more character development, but the laughs maybe aren't as quick and fast. Though, there's so many coming out of both countries that it's kind of hard to make a distinction between either one.
So would you say an Office is a sitcom or not really? Because I'm not sure how you describe that. Like the Office? The Office, yeah.
So I recently watched the American version, from start to finish, and it was great. I found it really, really funny. I will admit to attempting to watch the British version and being unable to.
Really?
Yeah, I just found it really, really awkward. And it kind of just made me cringe up internally. And
that is so fascinating. I found both versions to be cringeworthy, frankly. Hence, I never attempted to go past more than a couple episodes in each.
I found the American version loosened up a bit, and it got a bit more slapstick, a bit easier to digest, whereas the UK version was just slightly punishing to watch.
So we can blame it on Ricky Gervais, basically.
Yeah, let's blame it on Ricky. Yeah,
I think he can take it. He has the thick skin.
Yeah.
So as far as the differences between America and US, actually, one of the questions I was curious to ask you, since you've kind of dipped your toes in either world, what do you think is the similarities between let's say, research, since obviously you're a research scientist now, between doing research in America versus in the UK? Where do you see yourself more flourishing, if you will, and why?
Yeah, it's a difficult question in a way. I suppose the environments differ quite markedly in that I was, strictly speaking, in an academic environment during my time in the US, whereas I'm in much more of a biotech pharma kind of environment now. And I don't want to sound cliche, but there is the natural differences between the two. Where in an academic setting, you get fixated on a focal point of academic interest, and you hash that kind of research out to the bitter end, so to speak. And that happens in, obviously you get some support from your lab, but otherwise it's quite an independent, quite an isolated piece of work. Whereas in a biotech, pharma, industrial, what have you kind of setting, you're kind of, what's the word? Your environment for research is much wider, especially coming from a contract research organization like myself. You can be, dipping your toe in all kinds of different areas of biotech and drug discovery. So the focus is a lot wider, and I would say there's more of an integrated approach to the projects and to the research, in that because it's a drug discovery setting, there's a lot more stakeholders in the success and the progress of the project. So the teams feel bigger and they feel a bit more collaborative than perhaps what you would have got in an academic environment. They're not necessarily differences between research in the US and the UK. I would say one of the big differences in the US I found with SBP when we were working together, one thing that stuck out to me was the integration between biotech and what for us was an academic setting. So you had a real overlap between what was going on the Torrey Pines Mesa with lots of small startups and larger pharmaceutical companies. And a lot of that kind of bled into what we were doing at the SPP, which I haven't experienced as much in the UK. I've conducted academic research at CRUK Beeson Institute in the UK and some other institutions as well. And there's hints of that. So spin outs and collaborations with industry. But I don't know, with San Diego, it felt like that was taken to new extremes, and there was a real cross-pollination between the biotech sector and academia, which I think there's probably places in the UK that are similar, but I haven't quite had the same experience here that I did in the US.
So is this a result of maybe, let's say, I don't want to say like a class separation, but sort of a class separation in the UK when it comes to academic research versus kind of new breed of entrepreneurially minded people that are eager to try their hand in actually, doing business?
It's an interesting question. I'm not sure whether a class divide is maybe the right way to put it, though. Running with that idea, I do think that the wall between academia and industry can be a little more pronounced in the UK than perhaps, at least from what I saw in like California in the US. And I do think that's changing. There's a lot of, exciting initiatives coming up in my hometown in Newcastle and stuff like that, where you're seeing more available funding for academics who maybe want to transition to creating spin-out companies, or they want to engage more with industry. And perhaps that's what has been lacking in the past. Maybe it's that the money doesn't flow quite as freely between academia and industry as it does within those two groups. So they don't meet in the middle, so to speak. Though I think that could be poised to change.
Yeah. So just following in that vein, I know that there's specific types of grants in the US which sort of help. There's that kind of startup grants. So I don't think in that sense there is not such the same thing equivalent to that in the UK then, or you're not sure?
I'm sure there are, though exactly how they're administered, I'm not entirely sure, or kind of, how openly available they are. I think if you're an academic, I think kind of getting academic grants is probably a bit easier than trying to branch off into an industry or a spin-off venture. Though I have to say I've got no personal experience with that. That's just kind of from my peripheral speaking to people who've toyed with the idea. It doesn't sound like it's, a simple thing to do.
So I had a conversation with Peter Terreed from SVP last year, who also had who spent some time in the UK. And one of the things that we discussed with him on the podcast was the sort of the collegial spirit that is fundamentally, I would say, different in the UK versus the US, which something that we discussed with you right before we're recording this, where you can have like a share a pint with your mates, if you will. There's not the same thing, I think, in the US, which that's, for example, one of the things I really missed in my PhD time, which is being able to, on a Friday night, really experience going to a pub and bounce off some ideas.
I'll admit I missed it too when I was in the US. It didn't seem to be. I think the culture in the US was kind of like, you live and breathe the science, but you do that in the lab. At least that was the gist I got. Whereas I think in the UK, it's, you do your stint in the laboratory at the university, what have you. But then, as you say, you spill out into the pub, you have a couple of pints, you lubricate the mind, so to speak. And, a lot of discussion happens in those kind of environments in the UK. A lot of networking and a lot of, I don't know, I have a feeling like a lot of the more avant-garde ideas that sometimes end up sticking get dreamed up in pubs. I mean, I don't want to digress too far, but one of the best talks I've been to was Lord Robert Winston, who's a famous UK scientist, and he was talking about how he ended up producing one of the very first magnetic resonance images ever, because he was studying like ovulation in rabbits, I believe, and he didn't know how to measure what stage they were in their cycle effectively. And he was chatting to some guys at a pub in Cambridge or Oxford, the typical setting. Yeah. And there was essentially two physicists who he was talking to, and they were like, well, you'll never guess what we have back at the lab. So they kind of, went back to the lab half cut and put a rabbit in what was one of the world's first MRI machines. Wow. And I'm just like, it's almost a romanticized thing, but it does really happen.
Yeah. I feel on that note, it really feels to me that in the US, maybe I'm a little bit bitter in some ways about spending so much time in a laboratory. You spend so much time, working on your graduate research project or whatever, and you just are so mentally exhausted that you're just checking out and you don't really have enough time to socialize. And that, talking about science in a bar setting is not really a thing that you would necessarily seek out. But that's not how I felt when I was in the UK, for example. There was more of a defined structure to doing research. Maybe because I wasn't doing graduate school there, I was just doing an internship. But to me, there was like, you work and then you socialize and you can kind of implement that, bouncing off of ideas. And I heard not just, this is a great story that you just mentioned, but there was some other ones that I heard, perhaps, I think, both, what's his name? Now I'm blanking out. The guys that discovered the DNA, Francis Crick and James Watson, right? They were also in a pub where I think they shared in Cambridge their results for the first time, or so the legend says. So yeah, I completely see that this would be how it would go in that very romantic romanticized kind of way. So in that sense, what do you think will happen to these kinds of encounters going forward? I mean, I'm sure you miss it, just as much as I do, if not more. How do you think that's going to affect the research or, just how people interact with one another?
It is hard, that social aspect is often kind of sidelined, or it's not mentioned, but it's an important part of the scientific process, is being able to discuss things in an informal environment, perhaps without your supervisor watching and criticizing what you're saying, or just letting ideas flow freely. So it's hard to say. I think I've been pleasantly surprised with how the scientific community on the academic side and the drug discovery side of things has adapted, for instance, conferencing to be able to continue in a new form, much like what we're doing now, online in spite of COVID. So at least some of those discussions can go on. But I have to say, I've still found a lack of those kind of relaxed networking opportunities. You're not wandering around with a coffee, rubbing shoulders with your colleagues in different departments or different companies or different research groups. So who knows? Maybe there's a technological solution to it all, and we can do it all.
I hope so. Some kind of ale on tap through Microsoft Teams.
Hey, I mean, my partner's a postdoc, and they were, sent nibbles and wine in the post. So people are trying, to get these things, to get around the difficulties we're currently facing. But I don't think there's going to be a replacement for seeing people in person, and hopefully, given the progress on the vaccines and things like that, we can get to some sense of normality.
So when was the last time you were at a pub, like having a pint?
Don't make me.
I'm sorry.
Back.
So it hasn't been since before. Since the before times. It's
been a while. So we had a slight lull in the restrictions. So we're in our second lockdown. We had our first at the outbreak at the outset of COVID. Then we had maybe a period of 3 months where we entered a tiered system where we were able to, go back to some sense of normality for a short space of time before Christmas. Mm-hmm. And me and some colleagues, we were able to get out to a pub there because we had a reasonably regular meetup on a Friday, which, good to unwind and talk about what's going on in different projects and things like that. Mm-hmm. And I do miss that. But going back to your initial question, it's been a while since well before Christmas, I'd say.
Oh, man. Yeah. So hopefully your local pub is still not completely closed its doors. I heard that quite a few pubs across the UK have been basically just closed forever. And that's like, to me, like, I don't know, it hurts me, even though I have only very limited exposure to that. It still kind of feels like this like end of an era in some respects.
Yeah, I think the hope is that, things will bounce back in some respect. Mm-hmm. Once we get a handle on the situation, if we get a handle on the situation. Yeah, I think pubs, they're not the only ones. Any kind of business that relies on people getting out and socializing, whether it's live music or restaurants, they're all struggling. So it's all remains to be seen, unfortunately.
So which I guess leads me into your day-to-day activities. Now that you can't really go and socialize in a pub, you spend quite a lot of time working. I know you are a very busy person, and using this opportunity, I just want to say thank you for taking the time to speak with me. So being in a CRO, being a senior scientist at the CRO, I'm sure you have no shortage of people wanting to, ask you something, and there's like hard deadlines and things like that. But what is like maybe one thing that you get a kick out of working in this kind of, very fast-paced environment, and that you would not really find in any other, I guess, position?
Yeah, good question. I mean, as you know, I'm someone who gets bored easily, so the fast pace, and the variety of tasks that you might have to do on a day-to-day basis, that keeps me going. If I know that if one task becomes mundane, there's probably 5 more that I got to get my teeth into that require a level of, critical assessment, developing assays, asking scientific questions. So there's no shortage of that. In terms of things that I get a kick out of, there's freedom to, explore some of my personal interests in terms of areas of drug discovery or research that, I've built up an interest in over the years. And I like the freedom to apply those interests and, give something to the department. So things like I was involved with developing and screening drug combinations back at my time at AstraZeneca, and being able to apply that to my work currently at Sygnature Discovery, where I'm currently based, has been great. And I've got a lot of those kind of things in a back pocket, that you kind of build up over the course of your career, that you may not necessarily get to get your teeth into if you have a more focused, should you say, set of things to do. But as long as we're delivering on client projects and making sure their drug discovery programs are moving forward, there's always the opportunity to kind of incorporate new ideas, bolster the capabilities of the department and things like that. And seeing that progress, I get a kick out of that. It's fun.
So I think your work involves working with, I would say, basically like a cutting edge technology as far as laboratory automation, I would expect, and things like that. This is something that I'm personally very interested in having you maybe describe a little bit, like what's some of the things that you're seeing at the very end of this CRO world that you're working in. Where do you see this kind of I guess technology leading to, as far as how fast you can deliver on some of those projects, and how far are we in terms of this, just pace from the, let's say, beginning of the last year and all the way now? Like how far have we come in the last year, basically?
So I mean, pace is like the name of the game in drug discovery. I mean, your work has to be rigorous, it has to be considered, but time is always of the essence. It's about getting these treatments to clinic as fast as possible. Not often you're racing against multiple clocks, whether they be competitors or other things. So technology is a large part of that. I mean, we've invested in a lot of different assay platforms that kind of they're intended to make us more agile in the drug discovery process. So things that we do is we'll often run, functional assays and biophysical assays in parallel. So we'll be running multiple levels of drug discovery, bringing on expertise from cell biologists, biophysicists, biochemists, tackling the problem from multiple angles, because often you find if you can leverage that kind of resource at the start, pick the ideas that work best, then you'll get the quickest results in your drug discovery program. Another thing that we emphasize the importance of is interdisciplinary work. So we work very closely with our chemistry department and our DMPK department at early stages to make sure that, the chemicals we're testing, they're not going to be hampered by poor PK down the line, or, the chemists aren't going to come along and say, we can't do anything with this because it's a chemical dead end. So we're constantly talking to those guys so that we're not wasting time, when it comes to getting some kind of lead molecule in the pipeline later on. We don't want to hinder ourselves down the line. And that again involves a lot of different technology platforms. One of the most powerful that we've seen a lot of recently is fragment-based drug discovery, where we're taking really small 200 molecular weight chemical moieties, testing them in biophysical platforms. We get computational chemistry in a big way. So we're using virtual screening, computer modeling, looking at crystal structures of proteins, fitting things together. Coupling those kind of new techniques with more traditional screening lead-like compounds can really help. So I would, in a nutshell, it's if you're hitting the question from multiple angles and multiple techniques and multiple technologies, that naturally speeds up the drug discovery process, and it's something we put high value on when we're persecuting a drug discovery program.
It seems that there was so many different technologies, and as you say, hitting one project from so many different angles, the let's say activation energy, using biophysical terms, to understand how a certain type of molecular process works and how to influence that seems to be just increasingly more difficult as years go by, because we learn more, we have new different techniques to probe the same thing from all those different angles. I do think, is this the right sort of path to go? Because I think at some point we're going to hit the sort of, not the wall, but there's just going to be so much of different ways you can look at the same thing. When is the time to simplify things? I mean, that's a big and loaded question, but
yeah, but it's a good question. It's a question that's kind of on the mind of anyone who's involved in drug discovery. Is the scary question is, have we hit all the low-hanging fruit already? It's as you say, the cost in time and money for developing new drugs only increases, and there's got to be some component of maybe the easy targets. We've already got good candidates and good tool molecules against them. So it's an interesting question, because you're like, should we simplify it? But I wonder if the question is, should we be viewing things more holistically, to be honest, and rather than as you say, throwing resource in at the kind of trying to just find hits. Considering biological systems for what they are, which is complex, multi-component systems that are probably not going to respond to a single small molecule drug. And things have been moving that way for the best part of a decade, really. There's much more recognition that if we're going to use targeted therapies and realize personalized medicine, tailoring people's drugs to the type of tumor they have, for instance, that's probably not going to be one drug. It's probably going to be a cocktail, a combination of drugs, going back to what I said about some of my experience in the past. And even then, it may not just be small molecules. We've seen growing importance of, a big thing right now is vaccines, which we're seeing more techniques coming in. So employing the immune system with CAR Ts, immunomodulating, biologics, again, a hugely expanding area of drug discovery, especially in the oncology space, but also in the space of inflammatory diseases. I think what drug discovery will be is it's going to be building a toolkit of these things, and small molecules are only a small part of that. Mm-hmm. Then I think it's going to be a case of scientists in collaboration with clinicians, coming up with how you can really leverage all those different components against the disease in some kind of combination, whether it be small molecules, PROTACs, medicalized antibodies. I think at the end of the day, all these different technologies are going to, they're all going to be employed against the disease in parallel at some point.
Okay. I mean, so I think it's a bit of a wishful thinking to have all of this knowledge and all of these technologies to be at the back end of, I guess, of this kind of clinician or scientist of the future, where you could just really understand something quite simply, and then all of that body of knowledge just working in some kind of black box approach and just spitting you, okay, this is the combination of things that you need to add.
It's wishful thinking, but I think you've got to have an element of that, to be honest. You've got to imagine a future where we have some process that doesn't exist currently for matching a patient or a patient group to what the quote unquote ideal treatment regimen would be. And I think part of drug discovery, is making sure that once that capability is realized, that the treatments are available for patients' use.
Mm-hmm. So that's actually, this makes me wonder, the technologies that you use and the data that basically all of those instruments really provide you, do you really have a way to, let's say, store it in a way where you don't have to repeat the same thing as time goes on, where you have some kind of inherent database of understanding of, let's say, how your different moieties work for a certain protein target, for example, or similar target, like how much of the, let's say, stuff you need to repeat, or you can actually, really build up on that, because that's ultimately, I guess, the bottleneck. Can you keep up with building out those technologies that can store, and so you don't have to repeat the same things over and over again?
Yeah, I mean, it's no mean feat putting all that knowledge in one place, but we've got software platforms which at least go part way to doing that. So part of what I like about working in this kind of industry is it's no knowledge gets left behind. It's really rigorous. Everything's recorded, meticulously scrutinized, and it's logged because it's important to the client. It's important to the just general knowledge of the drug discovery department. Mm-hmm. So you never know when a piece of knowledge will be important down the line. And so in short, yes, we track everything, and we try to be mindful of things we've learned in the past. And we can always go back and pull out old information and apply new ideas to it. Mm-hmm. So I'm sure there's ways to improve upon that. Often the problem these days isn't producing and logging the data. It's processing the data and getting useful information from it, right?
Yeah, I bet. I think one of the things I've discovered is there's a whole other industry that supplies all those different software solutions for this data processing, so you don't really have this big silos of data that is just, you have really great quality data, it's all logged in perfectly, but it's only as good as how quickly you can retrieve it and put it to good use. So in your, I think, so as you say, you're working in CRO, and you previously had some industry experience as well. What do you think really separates CROs from working in a pharmaceutical company? Is there really a big distinction in terms of like day-to-day activities? Because I mean, there's quite a bit of overlap as far as, you have R&D departments, you have this drug discovery departments, but what is the one thing that maybe separates what you do in Sygnature versus if you had to do the same kind of stuff in some other company?
Yeah, it's a really good question. Yeah, face value, there's a lot of similarities. As you say, you're dealing with drug discovery pipelines, and a lot of the data production, data logging, and things like that. The culture of the way we conduct research is very similar to what you would see at Big Pharma. One of the key distinctions with a CRO is the client facing nature of it. So we work in smaller teams. We have a client who's a stakeholder for this work, and where essentially they're man on the ground, for better want of a phrase, persecuting their drug discovery project on their behalf, and that creates a different environment to what you would have in a pharmaceutical company, where you may have a whole department working on a single drug discovery project. You're more likely to be in a smaller team, and it's often the case, that you kind of integrate the client into that team in a way, and that project, that drug discovery project, kind of becomes personal to you, because that's like your baby. That's what you're doing for the on behalf of the client. So it feels a little bit more personal. You're not doing it on behalf of a big pharma organization. You're doing it. You almost treat it as though it's your personal project, and that ultimately translates into doing a good job for the client, because you have a vested interest in making sure that it all goes smoothly, and it's you ultimately get them that lead candidate at the end. But yeah, that client facing part of the role is quite unique to a CRO. That it's something you don't really find in a larger pharmaceutical setting.
Do you still feel the sense of like, I'm trying to help, let's say, patients at large, and how is that sort of relationship shaped as far as like in the company culture? You mentioned the client facing aspect of it, but how does maybe the eventual outcome of some work that you do eventually being part of the patient medication, like does that also permeate the work that you do?
Yeah, I mean, at the end of the day, that's why we do what we do. It's it's the guiding principle at the end of the day, and that's built into Sygnature Discovery's our value statement, that ultimately we're persecuting these drug discovery projects on behalf of the client in the hope and expectation that they're going on to make an impact in a clinical setting. And we've been lucky enough to see that happen on a number of occasions. We've had a number of compounds that we've helped persecute go into phase 1, 2, 3 clinical trials, and we've had clients give talks on successful translations from preclinical to clinical, and it's always heartening to see. That's what drives our efforts at the end of the day.
So would you say in a few years' time, when one of the compounds that you helped discover or finalize becoming a fully fledged drug, was that something that you personally hope to accomplish in some way, or is it just like, okay, that's nice, I'm doing it not just for that but for other reasons too?
Yeah, I mean, obviously there's multiple reasons, but it's never just, oh, that's nice, is it? It's a big deal if I've always got it in the back of my mind, when I'm seeing I'm working a drug discovery project, I'm running a functional assay, and I can see the compound becoming more and more potent, and getting better phys chem properties and better PK, and it works in cells. Maybe it'll work in in vivo models. The question constantly in your mind is like, is it going to work in a person? And you get a palpable sense of excitement when you see that drug come from just like, some poor, not very potent scribble in a chemist's notebook to, being this potential clinical candidate, and I long for the day where I can get all the way up to that end point.
Wow.
You really feel it. You are invested in these drug discovery projects, and you do get excited about them.
That's amazing. So now that you're in your CRO role, I want to take us one sort of step back. Obviously, before in a previous life, this is where we met. We met in San Diego, where both you and I were graduate students. So my question to you is, and this is what I would like to maybe call like a punk rock moment, where so you basically took a leap of faith in some regards, where you chose to essentially not pursue finishing the PhD and essentially return to UK. And basically, you're now a senior scientist at the CRO. Looking back, this has been almost 3 years now. I assume you're pretty happy with that decision. I can't see why you wouldn't be. But what is your, let's say, sense of like accomplishment? How do you feel really looking back at like that decision at the time? Did you see yourself being where you are now? And how was that decision really affect your life over these last few years?
Yeah, well, I mean, it was obviously it's a big decision, but you describe it as a leap of faith, and it's like I've got a habit of doing that, to be honest. I think it was a case of, moving to San Diego. That was kind of a big leap as well, because as you know, I started my my graduate program in Glasgow at the Beatson Institute, and transitioned to San Diego because my lab effectively packed up shop and moved to California. It was an interesting realization. I think perhaps spurred on by the fact I'd already had some experience in industry before. Mm-hmm. I started out immediately after my undergraduate studies working as a research tech with AstraZeneca. But in the 2 years I was in that pharma role, I was kind of always like, oh, I want to try big picture, blue sky science, go for a graduate program, do a PhD, and see where that takes me. And, it took me across the Atlantic, and it took me a bunch of places, but I got a palpable sense of when I was in that third year in the SBT that it was taking me somewhere I didn't really want to go. I got kind of the feeling that I'd hopped on the wrong bus. Mm-hmm. And I was like, really what I was wanting to do was it more of what I'd experienced in industry, to be honest, which is, persecuting these scientific ideas at the point of them becoming tangible therapies, basically. That's not to say I didn't enjoy my time in academia. I did some really interesting stuff, aging biology and epigenetics, which I still got a really big interest in, but I felt like it was it was time to kind of sidestep and think about what I wanted in terms of a career, and whether that was the right place to take me there. So it was a bit of a leap of faith, and it wasn't just professional reasons that I left. I had personal reasons for returning to the UK, but I would say overall it was a professional move. Mm-hmm. And did I see myself here when I essentially moved back to the UK? Kinda.
That's nice. Yeah.
Yeah. It's kind of what I was aiming for. So I'll tell you now. I got back to the UK. I wrote up my data as a master's thesis. Took a bit of time out to reorient what I wanted to do. I decided, I still want to work in a lab, just in a different context. So I, pursued career options in quite a wide range of things. I had interviews with a few startups that were doing interesting stuff in aging space, epigenetic space. I had a couple of interviews at places which were maybe a little more aligned with academic interests, but were kind of on the translational side. So moving ideas from academic labs into drug discovery programs, so to speak. I got some offers from that, others, not so fortunate. But then it was a case of this opportunity came up and it kind of ticked all the boxes. And, I got an initial interview and it seemed right up my street. And, the leadership at the company seemed great, really clued in. And I kind of got a feeling of, oh yeah, that's the culture I want to be in. So I'll go for that. And I think if I hadn't got the job at Sygnature where I am now, I'd have probably looked for something similar.
Mm-hmm. One of the things, maybe as we conclude this very fascinating conversation, because working in CRO is something I never seriously considered, just because of I was, I guess, in some cases hesitant to really go full steam ahead with tight deadlines and all that stuff. I'm curious, in this year that we live in now, not to say, this year is any really different than all the other years where we have had a really huge impact on the environment over the last few years. I made a resolution for myself as a New Year resolution to learn more about how scientific community has an impact on sustainable future for our planet. I'm curious, in your day-to-day activities, is there some part of you that wants to, I guess, have an impact not just on patients, but obviously on the way the business is conducted in a sustainable environment? I know this is a bit of a crazy pivot, but I'm curious, as we as scientists obviously have a role to play in driving this sustainable future, but in the company and like maybe you personally, like, do you feel like an increasing need to address that aspect of the business as well?
Yeah, you got it right. I think part of being scientifically minded is that you're aware of the ecological impact we have, perhaps more aware than the average citizen. Whether that's a blessing or a curse. I know it's hard as a scientist. We use a lot of disposable plasticware and things like that. But that's necessary to produce good science. And it's not to say that we can't do things to, address that. So it's funny you should ask the question. I'm one of the health, safety, and environment officers at Sygnature Discovery. So the environment part is something we take very seriously, and we do have initiatives at Sygnature to try and make sure that we're minimizing the impact we have on the environment with what we do. Because that's an important thing. We've got to look after the planet as much as we look after ourselves. It's cyclical, really. So we try our best to put as much plastic back into the recycling system as we possibly can. And we're always keen to, reduce packaging and make sure that we're using reagents or techniques that produce less waste where it's scientifically prudent to do so. And also just in day-to-day operations, we got rid of plastic cups a couple of years ago. For Christmas, we all got metal, drinks bottles, because, every little helps when it comes to this kind of thing. And, we're always open to ideas on how to minimize our impact on the environment in general.
So do you think it's the lack of, let's say, available recycling technologies, or is this just a pure research necessity currently that sort of precludes, organizations such as yours and many others to really make a significant leap forward in this regard?
It's multiple things, isn't it? There's a whole system to consider when it comes to the kind of pollution, for want of a better words, that we produce and what we can do with it. One of the questions is, could we recycle contaminated waste? I don't know. Maybe there are processes that could come in the future that would allow us to safely decontaminate laboratory waste so we could use it again. Mm-hmm. Could we use different materials for certain aspects, like pipette tips that could be reused? I'm not entirely sure. I know some of the things that we do, we'll use things in lieu of pipette tips for dispensing compounds, because pipette tips are a waste. Mm-hmm. They're just disposable plastic. So we'll use metal tipped automated dispensers to get around that. Or, something we use 90% of the time these days is we use contact-free acoustic dosing platforms, mm-hmm. Which cut down on a lot of plastic waste. So that's an interesting question.
So would you say, just in the hypothetical world, if you had a company that does something in that sort of, let's say they have a technology or a way to minimize waste or mitigate the effects of it, is it something that, let's say, you in a company would be like, okay, we're interested in maybe exploring that as a pilot option?
Yeah, without a doubt. We have quite a good system where the staff at Sygnature, they're always kind of encouraged to suggest ways in which we can improve on our environmental impact and things like mm-hmm. that. So a lot of our current initiatives have come out of that. Likewise, if there are, as you mentioned, more like institutional or commercial endeavors which would allow us to reduce the waste we produce or the energy we use or things like that, yeah, that's something that we would certainly consider. I'm sure.
Mm-hmm. Well, yeah, I just feel like as this year rolled in, it just became ever more palpable, because if you recall, maybe early on in the COVID time, it even feels strange even saying these things. I remember reading all these reports, whales in the Hudson or something like really crazy, where things are slowing down. You see nature sort of taking back what it's lost, and it just sort of made me realize even more, prominently just how much of an impact we are having. And if there is a way to keep that conversation evolving, even in this kind of instance, where this kind of might get exposure, that there's companies that are interested in exploring new technologies, every little bit helps. And it's nice to see that even places with such strict, let's say, deadlines, strict demand for all the scientific rigor, as you mentioned, there is room for improvement. There is always room for innovation.
Yeah, no, I completely agree that, it's a topic we should be bringing up in conversation more frequently, and hopefully in conversations where it wouldn't usually crop up. I think in research, it's as I say, it's an even more poignant conversation, because we do produce a lot of waste. We do have to be mindful of the kind of waste that we're producing. And equally, I'm sure there's a lot of other industries that would benefit from the same conversation. And I agree, it's, in a way, this year started out with, the fires in Australia and things like that. And it brings home kind of what kind of impact people are having, and what we can do to try and mitigate that. So I do think it's an important conversation.
Yeah. Well, Adam, thank you so much for having a chance to speak with me today. I really appreciate it. I look forward to having a pint of Newcastle ale, and hopefully that is not virtual in the not too distant future.
Our fingers are tightly crossed. Thank you for having me. It's been a pleasure.